I had to do that so you can see me. Minister, members of the board, director, ladies and gentlemen, it's a real pleasure to be here, particularly on the 30th anniversary of the Cyprus Institute of Neurology and Genetics. It's the first time for Anna and myself to visit Cyprus, but it's one of the countries we've wanted to come to for many, many years. I went to the Institute today. It was a very impressive experience. What I saw was a well-equipped laboratory full of skilled and motivated researchers tackling important problems for society and for the country of Cyprus. As you heard, it's about to be expanded. This is a very good development for Cypriot science and also for the people of Cyprus. Well, ladies and gentlemen, I also come from a research institute. It's called the Crick Institute, and we have an interesting logo. This is the most interesting slide that I'm going to show you. Look at that. Look, 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 bang. And this is the Institute. It's located behind the British Library in central London, next to St Pancras Station. It's very large. It's 100,000 square meters. We have 1,300 scientists there. I'm the founding director, and I run a laboratory there of 6 or so graduate students, a couple of postdoctoral workers, and I'm going to tell you tonight about the research from that laboratory. But because I know not everybody here is a research biologist, I'm going to give a little introduction as to why the problem we're studying, I think, is interesting. I'm going to talk about cells. We are all made of cells. It's the basic unit of life, the basic structural unit, the basic functional unit of life. It's very important, if not least because maybe I should remind all of us that we were all once a single cell. All of us. So you should be interested in cells, is what I'm trying to say to you. My particular interest is how cells reproduce themselves, going from 1 to 2, because it's the basis of all growth and development. This process of reproduction is called the cell cycle, and during the cell cycle there are a number of events that occur in a particular sequence, in a particular temporal order, that are necessary for the successful reproduction of cells. There are 2 major events which have been central to my research all my life, which are key to the cell cycles in all living eukaryotic organisms. The first event is called S phase. That is the period in the cycle when the DNA is copied or replicated. That leads to the duplication of all genes. Second key event is called M phase or mitosis, and that's at the end of the cell cycle when the chromosomes, which contain all the genes that were copied in S phase, become segregated apart and distributed in the 2 newly divided cells so that each cell receives a full complement of the genetic information. In other words, this process is key for heredity, it's key for inheritance, and of course is a crucial part of genetics, which the Institute studies. My life's work has been committed on how that process of the cell cycle is controlled, how the temporal order of S phase and mitosis is maintained. It's a very basic problem, a fundamental problem of biology, but although it's basic, it's actually very relevant for the application of science. For example, you cannot understand diseases like cancer without understanding how the cell cycle is regulated. And I mention that so I can make one other point before I start talking about my research. And that is that research, scientific research, is a spectrum. It ranges from discovery basic research through translation of those discoveries into potential applications, and then the applications for societal good, for health or generation of wealth, for example. Sometimes there can be too much emphasis in society on translation and application because it's seen to benefit society immediately. But without knowledge generation, with discovery research, soon they will end up with nothing that can be applied for societal good. This is obvious but can be ignored. One of my predecessors as a president of the Royal Society, the Academy in the UK, George Porter, he was a chemist Nobel laureate, he very nicely put it this way. He said, ignoring basic discovery research is like building a tower ever higher whilst ignoring the foundations. It is only a matter of time before it all collapses. So what we need is a broad spectrum of research from discovery through to application if science is to serve properly society. Now, I now turn to my research project, which is, as I said, basic and discovery, which is around how the cell cycle is regulated. And I'm going to begin by briefly describing what you might call the textbook view of how the control occurs. It has to be said that this textbook view has been influenced quite significantly by research from my own laboratory. But I want to now, as my talk develops, give a somewhat different perspective upon that textbook view, a different way of thinking about the problem, because science evolves. And although a Nobel Prize might have been given for this, that doesn't mean it stops there. It can continue. So what is that textbook view? Well, it's shown here. This is a nice book called The Cell Cycle by David Morgan. There you see the various events of the cell cycle. Notice in purple, S phase. Then we have mitosis in green at the end. Now, the fundamental control there is a family of protein kinases. These are enzymes that add phosphates to other proteins, changing how those proteins work, and they often work as switches in the way that cells, and for that matter, all other biological functions work. These protein kinases, as you heard in the introduction, are called cyclin-dependent kinases, or CDKs for short. You're going to hear a lot about this type of enzyme. My talk is going to be based upon it. Now, the traditional textbook view is that there's a range of these cyclin-dependent kinases, a family of them, 4, 5, 6, depending on different organisms, and that these CDKs have different substrate specificities. In other words, they phosphorylate different proteins, and these proteins are required at different stages of the cell cycle. So you see up here, a G1S cyclin-dependent kinase at the beginning of the cycle is necessary to enter into S phase. An S phase cyclin-dependent kinase is necessary to undertake S phase. Then towards the end of the cell cycle, an M phase CDK is necessary for entry and progression through mitosis. A cyclin-dependent kinase is made of two components, a protein kinase and a cyclin molecule. And if any of you heard Tim Hunt give the lecture that when he visited here, he worked and indeed was the first discoverer of the cyclin component of this particular control. So the model is that different protein kinases, CDKs, appear and disappear as you go through the cell cycle. They phosphorylate different proteins that go up and down through the cell cycle to bring about all these different events. And that is the basis of the temporal order of the cell cycle. Now, this is subject to a lot of regulation. You see the CDK in the middle there? Just put your eyes round all arrows there, which are all the different types of regulation that have been proposed. Now, those biologists who write reviews of areas like this often try to put everything together to produce a sort of map of how it all regulates. And this gives you an example. Now, I hope you can't read any of the names here, because I don't want you to read them. I want you to look at this diagram and feel vaguely depressed. And I want you to feel vaguely depressed because it looks far too complex to understand. And you would be right to think that it is too complex to understand. And one of the things that disturbed me in the last 10, 15 years is that we identify more and more biological functions, usually proteins, that are involved in a process. And we think we understand them if we list them and connect them together. And what's happening is we are describing the problem, but we are not understanding the problem. And this is really going to be central to what I'm talking about. Because faced with this, working with a very simple organism called yeast, I wanted to see whether we could simplify this network, because it looks a little bit like the underground map in London, I'm sure you'll agree. By simplifying this network, could we remove functions and yet still get the process to work reasonably well? And if we could do that, we would then focus on what really matters rather than what might matter or only matters a little bit. And so that's really central to my work in this area in recent years. Now, I've been trying to sell this project to new graduate students and postdocs for some years, and nobody was interested till this man came along. This is Damien Kadrusa, and he was a bold man. He liked some risks. He tells me that's him on that board up there. I'm forced to believe him, I have no idea. You can see him down the right-hand corner here. And I gave him the challenge of seeing whether he could simplify this control network. Now, this is the cell cycle. Look, you have an S phase and mitosis, G1, G2. And this is in the fission yeast, Schizosaccharomyces pombe, which we work on. I'll show you a picture in a moment. And what I want to show you in this mitotic cell cycle is that there are 4 different cyclin-dependent kinases. Start at the bottom left. CDK1, that's the protein kinase, it's the same in all 4. But PUC1, SIG1, SIG2, and CDC13 are all 4 different cyclins that we postulated had particular roles in these different phases of the cell cycle. So I gave him the challenge, how can you simplify this? And what we started was, well, what we could do is make a fusion protein between the CDK and one of the cyclins, because that would remove some of the controls. And so we make a single protein rather than having 2 proteins that have got to find each other and make complexes. And would that work? So we started with that, trying to simplify some of this regulation, fusing together the CDC13 cyclin with the CDC2 protein kinase. And because we could do gene editing in fission yeast ever since 1980, this is what, when people talk about CRISPR-Cas that is now used in mammalian cells, in yeast we could do the same thing for the last 40 years. In fact, my lab discovered it for fission yeast in 1980. We could create this fusion protein and put it into a fission yeast and replace all other genes that were relevant with that particular construct. So what we did is fuse that protein, fuse the gene that encoded this protein, and put it in the genome. And this is what the cells look like. This is your first glimpse of fission yeast cells. They're very simple. They look like sausages that divide in the middle. And what I want to show you is on the left is a normal wild-type cells. Nothing's happened. In the middle, what you see is a strain where we've deleted CDC2, the gene encoding CDC2, deleted CDC13 and replaced it with a fusion protein of CDC13 linked with L to CDC2. And you see these cells are growing perfectly normally. For those interested in Western blots, because it's fusion, you'll see the protein is of a higher molecular weight. Now, Damien, when faced with that, thought, well, if that works, why don't I get rid of all the other cyclins and see if that works? Move to the right-hand slide, and you'll see he then marched through the genome and deleted SIG1, SIG2, PUC1, and it still works perfectly well. Now, I hope you understand what's happened here. We have deleted all the cyclin-dependent kinases except one. And that one is sufficient to drive the entire cell cycle. And this was not expected. Because it wasn't expected, we tested it more thoroughly. So what we did is introduce mutations into the CDC13 cyclin component, making it temperature sensitive, so it wouldn't grow at high temperature. And in the CDC2, what we did, and I need to explain this, we made a mutation in the ATP binding site. ATP is needed for the protein kinase's activity, in such a way that it was increased in size so that a bulky ATP analog, a bigger ATP, could actually get in there and inhibit that protein kinase. This meant we had a specific inhibitor just of this one protein kinase, because there's 150 protein kinases in fission yeast, 500 in our cells, and we only wanted to inhibit this one. This is this experiment here. The controls are the top line, and you can see we stained nuclei in the far right-hand figure there. In the middle one, we're at low temperature, but we've put in that bulky ATP analog. Look at the cells in the middle, see how elongated they are, because although they can grow, they cannot reproduce, so they just keep growing. Look to the right and you'll see they accumulate with a single nucleus. If you look at the DNA content, some of the nuclei have an unreplicated amount of DNA, some are replicated. So this means that the CDK element here is absolutely required for cell cycle progression when it's present in this chimeric protein. The bottom one is exactly the same experiment at high temperature where we knock out the cyclin. So we concluded from this that you only need one cyclin-dependent kinase to complete all the events of the cycle. This overturned the work that we'd done ourselves and others had done over the years, which thought all of them were unique and special and required. We then turned our attention to the meiotic cell cycle. And this was carried out by a postdoc in my lab, Pilar here, who came from Spain. And she investigated the cell cycle in the meiotic cell cycle. That's the cell cycle that makes the gametes. In our cells, it would be sperm and an egg. In fission yeast, it's single-celled. And we identified, and when I say we, I meant Pilar identified, that there were two other CDKs required, one encoded by CRS1 and one encoded by REM1. She investigated their roles and showed that they had some function at these different steps of the meiotic cell cycle. And then we asked the question, can we now get rid of all these further to CDKs and drive it by that single fusion protein? Now, this is an experiment we show here. It's complicated. I perhaps don't really want to take you through it all. All you need to know is for meiosis to be successfully completed, you have to get 4, you see the black blobs are nuclei, you have to get 4 nuclei in each cell, which is the product of meiosis. And what she showed is that she could delete all 6 of those cyclin-dependent kinases and replace them with the fusion protein as long, notice the number 4, she simply had to put 4 copies of the CDK in, up the expression. So as long as you up the expression, you could replace it all with one CDK. So this was our conclusion. that there's 6, just look at the bottom line. A single cyclin-dependent cyclin can drive both the mitotic and the meiotic cell cycles. And this, of course, overturned the traditional view. Now, it also led, how could that work? How could you bring about S phase, mitosis, mitosis, with just one cyclin-dependent kinase? Well, we had a model, and it's a very simple model. Here we have the cell cycle on the left. The intensity of redness is the amount of CDK activity you have in the cell. You start in G1 and it's white, there's no activity. As the cell proceeds through the cell cycle, the red gets stronger, and when it's light red, you get S phase. When it's a darker red, you actually inhibit S phase. That explains why there's only one S phase in each cell cycle. And when it gets to mitosis, you have a high amount of activity, and that brings about mitosis. So what matters, we suggested, was not qualitative differences between the 4 or 6 CDKs, but just the absolute level of CDK activity that was present in the cell. I've rolled out the cell cycle in the diagram on the right. What we have is CDK activity going up in this sort of seesaw, initiating S phase, blocking S phase, and initiating then mitosis. Then with cyclin being destroyed, demonstrated by my friend Tim Hunt, the whole activity falls to a low level and it starts again. This couldn't be a simpler model compared to the slide I showed you at the beginning of my talk. This is very simple, and it's testable. Because we could use that mutation. Do you remember the one I told you where we make the ATP binding site bigger and we can add an analog? You may remember that. So what we can do is add that to living cells. And what this model would suggest is that to undergo mitosis, you need a lot of activity. So you can inhibit that with a small amount of inhibitor. You require such a small amount to undergo S phase, you need a lot of inhibitor to block that. And then interestingly, what happens in the middle? If we believe this, as I've written it here, it means a cell in the middle there, in the light pink. If you let the activity, if you're arrested there, if you let the activity rise, you go into mitosis. If you force it down and then let it rise, you reset the cycle so it will actually undergo S phase. And so those were the 3 experiments that we wanted to do. What that last experiment would show is there is no arrow of time in the cell cycle. You can go backwards and forwards simply by manipulating the cyclin-dependent kinase activity. This is the first experiment. Cells are synchronized in G2. They are then released into different levels of that inhibitor, ranging from 25 nanomolar up to 300 nanomolar. This graphs the numbers of cells that go into mitosis. What you can see with the black line, that it goes into mitosis about 45 minutes after you have released the cells. And you can see that if you add increasing amounts of inhibitor, that you delay, first of all, the peak, and then you eliminate it altogether somewhere between 100 nanomolar and 300 nanomolar. All I want you to remember is you need between 100 nanomolar and 300 nanomolar to block. That's 0.1 to 0.3 micromolar. Then we look to see how much inhibitor was needed to block S phase. This is done in a different way. We've also synchronized cells, but we use a fax machine to measure whether cells have replicated their DNA or not. And what you should concentrate on are just the black squares, because the black squares are the proportion of the population of cells that are blocked in an unreplicated state. If we add no inhibitor, we have no 1C cells, they go immediately into S phase. That's the DMSO experiment up there. If we add 1 micromolar, you'll see black squares go up, we get a short delay. We add 2.5, the delay is longer. We add 5, it's longer again. If we added 10, I don't have it in here, it's another 30, 40 minutes longer. What you will remember is we could block mitosis with between 0.1 and 0.3 micromolar. To block S phase, we need 5 to 10 micromolar, 30 times more. So this tells us that we need much less activity to go into S phase than mitosis. This is the third arrow of time experiment. What we did here is arrested cells in G2 with 1 micromolar of inhibitor. If we wash that out, cells go into mitosis. But the interesting experiment is on the right. We arrest them in G2 with 1 micromolar. We add 10 micromolar, so we inhibit very strongly. Then we wash that out and let it rise again. And this time they go into S phase. Do you see on the FACS analysis how they undergo replication? So this means that we have shown that the experiment, and I can jump over that to here, that this experiment works. In other words, it's the quantitative level of activity that determines whether you go into S phase or mitosis. There is no arrow of time. Where you are in the cell cycle depends on your CDK activity. Now, because this is radical, we thought we should then look at the effect this had on the substrates that are phosphorylated by CDK. And this was done by a graduate student in my lab called Matthew Swaffer. He's shown here in New York with a collaboration with our proteomics unit in the Crick. So this work has all been done by my graduate student with the analysis of the technical core. Well, what he did, let's leave him up for a moment and I'll describe the experiment. What he did was to identify substrates phosphorylated by the CDK, and he did it in a very rigorous way. What he looked for was what we call the consensus site of amino acids that become phosphorylated by CDKs. And then he went on to show that if you inhibit CDK activity, then these phosphorylation sites become dephosphorylated. And so we know that they are definitely dependent upon CDK activity. And doing this, he identified between 250 and 300 targets. And the results I'll show you are a combination of those particular 250 or so substrates. And the first thing he did was to look at those substrates required for mitosis and those substrates that are required for S phase. And we grouped them together, and what you see here, as cells proceed through the cycle, those S phase substrates become phosphorylated, that's the light blue line, right at the beginning of the cycle. Whereas the ones required for mitosis are not phosphorylated till the end of the cycle. Interestingly, the substrates that are required for S phase, they become phosphorylated early, but they don't become dephosphorylated. They simply continue to get more and more phosphorylated as you proceed through the cycle, and then they all drop when the cyclin is degraded. So this again is not what the standard model predicted, which is waves of phosphorylation as you go through the cell cycle. This experiment had never been done to this level of resolution for people to conclude this. Again, to try and see what was going on, what Matthew then did was to titrate in increasing amounts of the inhibitor. That's this NMPPP1, and you'll see that this is a log scale with nanomolar amounts. And what you can see is to inhibit the phosphorylation of S-phase substrates, that's the light grey line, you require something like 3,000 nanomolar. Whereas to inhibit mitosis, you only require substrates, you only require 1 to 200 nanomolar. So this actually gives you a biochemical readout of what I've already shown you in terms of cytology. For those who really are interested in protein kinases, and I recognize that's not everybody in the audience, I'm going to show you one more figure here, which is a measure of protein kinase activity in vivo. Normally when you measure enzyme activities. You take cells, you break them open, and you do an enzyme assay in vitro. And I've always wanted to do an assay on living cells. And what we did here was to take our cells being driven by the CDK that we can inhibit with the inhibitor, add a lot of inhibitor, the substrates become dephosphorylated, and then we wash the inhibitor out and monitor the rate at which those substrates become phosphorylated in living cells. We haven't broken them down, we're phosphorylating, measuring the phosphorylation in living cells. And we can do it at different stages of the cell cycle, and we can do it with substrates that become phosphorylated in the beginning of the cycle and the end of the cycle. Look at the codes here. Look at top left. G1 cells, S phase cells, G2 cells and mitosis, you can see in vivo activity is dramatically different as you proceed through the cycle. I've done a short time course at the bottom there, and you can see that the activity increases 50 to 100-fold through the cell cycle. That's a huge difference. Yet, the low activity down at the S phase is still bringing about a biological output. So protein kinases aren't either on or off, as is often discussed. Their level is like a rheostat. At low levels, you get an outcome. At higher levels, you get other outcomes. And then you can see the early substrates, the substrates that are phosphorylated in the middle of the cycle, and the late ones. The late ones are phosphorylated in vivo, in this in vivo assay, at a much lower rate than the early ones. Again, fitting in with our model. So this is where we are. We can drive the whole of the cell cycle with a single CDK. In the middle there you can see that we phosphorylate early ones early in the cycle, substrates, ones in the middle, late ones at the end, and on the right we have our rheostat model. As CDK activity goes up, we phosphorylate substrates required at different stages of the cell cycle. So the whole system is working by the level of CDK activity and not a range of different CDKs bringing about these events. Now, I'm going to summarize, I'm coming near the end of my talk. I'm going to summarize the problems we are now studying with this, and I will show you, as there are many geneticists in the audience, the genetics we're trying to do to get at that problem, and then I will give you a final summary. What we are interested in is how this wide range, 50 to 100-fold, of CDK activity is regulated, because this is a very wide dynamic range. We are interested in how that activity is determined by the size of the cell, because these cells get bigger as they go through the cell cycle, and that regulates activity. We're interested in how it's determined by the ploidy of the cell, because when you have a diploid or tetraploid, the cells are twice as big when they undergo mitosis. And I'm also interested in thresholds, how you can turn an analog increase in CDK activity into a digital outcome of developmental events. Now, to get at these problems, we have to understand all the gene functions that we need to be thinking about. So I'm going back to the complexity, the things we threw in part away, just to see whether some of these influence these things. And we did 2 genetic screens that I'm going to describe to you. In the first one, we based it on the fact that we had fission yeast, there's 5,000 genes, and we deleted each one of those genes one at a time. So we have 5,000 strains, each one of which is deleted with one gene. This is the most comprehensive deletion collection available in a eukaryote. It's bigger than in budding yeast, for example. And we have an arrayed collection of heterozygous diploids deleted for these genes. We can sporulate them and make haploids which are deleted for any gene that we wish. This was constructed by a consortium led by my lab and based partly in South Korea. Jackie Hales, who's recently retired from my lab, put that together and did a screen that is described here, a haploinsufficiency screen. Now, the geneticists will know what this means, but I'll just briefly describe it. What it means is this. that if you take a heterozygous diploid, where only one gene is wild type and one gene is deleted, most of the time you reduce the amount of protein made by that gene from 100% to 50%. Not all the time, but most of the time. This means that if you see a biological consequence, a readout, and remember we're interested in the cell cycle, but if you see an impact on the cell cycle when you've reduced the protein level from 100% to 50%, then that is suggestive that maybe that gene may have a rate limiting influence on that process. Often you only see an impact if you reduce activities to 1 or 2 or 5%, but in this case, if you see a reduction from 100 to 50%, you think it may well be important. That's described here. What we looked for are cells that, when reduced to half the level, either advance cells at a small size into the cell cycle or delay them from going through the cell cycle. This led, because we look at the genome deletion, to a collection here of nearly 20 genes. Now, we had been studying the genetics of fission yeast for the cell cycle for over 30 years, and we had only identified by classical methods 30 to 40% of these genes. So these genomic approaches that we were using there identified a much fuller complement of genes that we needed. In particular, when we grouped them together, if you look at the top right corner over there, a total of 7 of those genes were involved in nuclear cytoplasmic transport. This had never been taken seriously in the field as being a key factor in control, or at least not very seriously, but the fact 7 of our 18 or 19 genes were involved in that process was very telling. If you look to the left, you'll see all the CDK type genes, CDC2, CDC13, were also identified by this screen, which validated what we'd done, and it included other genes that we'd not at the time thought were important. So that was one screen. The second one was to take the genome deletion collection and ask, when a gene is deleted, this time in a haploid, which cells are advanced prematurely into mitosis. If you look here, we measure that by cell length. On the far right, you'll see wild type. On the far left, you'll see WEE1. This divides at about half the size. I should mention I discovered this gene in Edinburgh in 1975. I called it wee because that's the Scottish word for small. I used to think it was funny, but 30, 40 years later, the joke gets a bit thin, okay? But it is the most extreme of the strains, but because we are looking at deleted strains, we could screen the entire genome, and we took a cutoff of 10% smaller, and you can see in the pictures of the cells, the ones on the left are obviously advanced, the ones on the right are normal, and you see the other ones are intermediate. Now, some of these effects are quite small, and people didn't believe them. So what Francisco Navarro, another Spanish postdoc in my lab, did was to make double, triple, and quadruple mutants. And I just want to show you this. Here on the left, we have wild-type cells. On the next one, we have deleted 2 genes, ZFS1 and SKI3. Next one, 3 genes. Next gene's 4 genes. Next gene's 5. And you'll see the effect gets bigger and bigger and bigger. All of these genes have some effect. We don't know what they do, but they are all candidates of having something to do with the regulation. I put all these together. These are the genes. There's about 30 genes that we have to think about, and 3 of those, WEE1, POM1, PPA2, were identified in both the screens. So what we've done is, with this analysis, we've identified all the cell cycle genes. I didn't mention that. There's about 500 of those that are required for the eukaryotic cell cycle. And we've identified about 30 of them that we have to think about in terms of regulation. So, summary. I've shown you, I hope, that rising CDK activity is the basis of order through the cell cycle. I didn't really show you, except very briefly, that some of the components there are the CDK core regulatory element, nuclear cytoplasmic transport, chromatin also. We've done more work which I didn't have time to tell you about how cell size is measured, how cellular localization is important, and how positive feedback controls are operating. None of these studies are completed, but they're all work that are actually in progress. Let me go back to this London Underground map. Now we've done this study, I want to suggest how we should perhaps think about complex biological functions like the cell cycle. We need to collect all the genes that might be having an impact. We did that in this genomic approach that I just described to you, deleting all the genes in fission yeast. Then you have a list of the parts that you should think about. Often people stop there and they create an interactome like this and they go no further. You then have to do really hard work taking this apart, and what we described was deleting a number of genes, all the other cyclins, we deleted others as well that I didn't explain to you, to work out what really matters in the regulation. That revealed to us that what really mattered was the quantitative level of CDK activity and not the qualitative differences. It revealed other things I've not had time to explain to you. And then you lead to better understanding. So what you have to do is describe the complexity and then reduce the complexity to a point which it leads to understanding. It sounds complicated, but you cannot just describe complexity and then think you've solved the problem. You have to take it on to understanding. Well, this is the Crick Institute. This is St Pancras Station there, the Eurostar terminal, King's Cross behind it. Do you see this? That's the Crick Institute. Please, I know some of you have visited, but please come. You're very welcome. And there we have a romantic view of London in the background. And this, of course, is my logo, and you know what I'm going to do now, don't you? Oh, yes, it might work. Let's see. So it's the end of the talk, and therefore the logo has to disappear or almost disappear. There it goes. I'll leave you with this. Thank you very much. Thank you.